Welcome to our Microbiology Journal Club here on our Monday at 3 p.m. Today we're going to be talking about this paper, which appeared in Nature Microbiology, and I'm not going to try to read that one through the mirror thing that I did last time. But this is Evolution of Atypical Enteropathogenic E. coli by Repeated Acquisition of LEE Pathogenicity Island Variants. So there's a lot crammed into that title, which we'll start to unpack just as an introduction to get everybody up to speed on what are we talking about with all these various types of E. coli. But before we get into that, I wanted to give everybody an opportunity to introduce themselves. So I'm Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island. And I used to work on plasmids in E. coli and Salmonella. So this is really interesting to me because one of the things we were thinking about was how these plasmids might facilitate horizontal gene transfer of things like virulence determinants. So I really liked reading this paper. I think it was a very good suggestion. And I'll ask Karen to introduce herself next because she's right next to me in the little panel. So Karen, would you like to introduce yourself?
Hi. Okay, everybody can hear me. Hi, my name is Karen. I'm a PhD student at Simon Fraser University in Vancouver, in Canada. And I work on a bacteria called Francisella tularensis and its associated virulence factors. But my interest in this paper is really interesting because our lab also works on E. coli and specifically EPEC and EHEC, but we work more on the host side, so I'm more of a cellular microbiologist in that sense. And we work on, but we do a lot of things with TEER and intimin mutants, and so this was really interesting for me to read, even though I don't actually work on it, but to kind of understand the context of all the other things that are going on in my lab as well.
Oh, fantastic! That's great. I didn't realize you guys covered that in your lab as well.
Yeah, we do. Yeah.
That's awesome. Very cool. So you get to be our virulence determinant expert in this paper.
I'll try my best.
Okay, so Joel, you're next to Karen and my panels. Would you like to go next?
Sure. I'm Joel Graff. I'm an assistant professor at Montana Tech in Butte, Montana, and this is my second journal club. So Grace and Karen are new to me, but looking forward to this. This paper's a bit outside my comfort zone. My background's in virology for my PhD work, and then in parasitology and macrophages for my postgraduate work. I think I understand more or less what the paper shows, but I printed it off in black and white, and so Laura has figured out how to work the screenshot. So I'm looking forward to seeing these figures in color because I think that'll make a lot more sense. And I'll just play the role of ignorant but curious question asker.
Yeah, please do, because I think that, I mean, I have not even a foot, I'd say like a couple of toes into this. So I think it's good to have the perspective of people who may not have a background in this. So yeah, please stop us for explanation at any point. And Grace, would you like to introduce yourself? Unmute yourself first.
Sorry, I didn't want to make too much background noise. I met Laura at a workshop with Pat Schloss, learning how to program Mother. I'm a wannabe programmer, but I have some grand, visions of something I'd like to compile. But I'm very interested in papers like this, although I don't understand 99% of it. So I'll be really gleaning a lot of information from what Laura reviews and comments actually, but what I see is actually from the clinical side. I'm a clinical pharmacist, and I practice functional medicine as a practitioner, and we use different functional medicine labs, integrative labs. I can send you some examples of what I see, but the labs culture stool, and then they use MALDI-TOF to quantify and identify the strains. So it's not ideal to look at E. coli, as you know, and they are able to somehow, I don't know how they differentiate exactly from the technical side, but they differentiate mucoid E. coli, the hemorrhagic ones, and adhesive E. coli on the testing. But it makes a kind of difference a little bit to how we approach things clinically. and to rebuild the biome and like try to squeeze some of the toxic E. coli out and launch in, you know, newer probiotic E. coli. There's like a lot of different tools, like probiotic E. coli, Nissl strain, and then some phages that are targeted. They're over the counter and they're E. coli cocktails, and they target specific toxic E. coli, I guess strains like this. But I actually normally don't see the strain that come up on culture, just the mucoid and other toxic ones. So I've read some papers where there's covirulence with yeast. And yeast, as you know, if there's an overgrowth, they can even go into macrophages. So that might be interesting in your background. They go into the macrophages and become kind of intracellular pathogens. So I'm kind of just interested in all the ways the gut becomes pathogenic, the microbiota becomes pathogenic instead of helpful for the host.
Yeah, great, thank you. And I think that's an interesting perspective to have, especially for this paper. And I just saw Dave pop up. So Dave, we're wrapping up introductions, so that was perfect timing. Do you want to give a quick introduction?
Yeah, I'm a very tired Dave Baltrist. University of Arizona, microbes, and failed transposon experiments in microbiology labs today. So.
Did you just say failed transposon experiment?
The strain, I started it last night, and it just hit like an OD of like 0.1, when usually it's like saturated by now. So.
Oh no, yeah.
So I just had to switch everything to like 10-to-1 and matings, and we'll see if it works. It should work, but.
Yeah. This is, I think this is the thing that my research students realize very quickly, is that you kind of think science should be very orderly and you do these things, and you're like, no, most of your day is spent troubleshooting the things that
we're just making stuff up. This has been an extra bad term, though, because I'm way scattered in like 8 different directions, and so if, you know, I'm prepping everything, I'm just, you know, you grow something on the wrong plate accidentally and it sets you back a couple days, and that just doesn't work when you've got a lab to do, so.
Yeah, yeah. No, that's true. That's true. All right, well, in the midst of everything you're doing, thanks for joining us.
No problem.
Okay, so this paper, I really liked this paper. You know, Nature Microbiology, I'm going to say off the bat here, I really wish it was open access, because they're publishing some great stuff, but it's apparently a giant pain in the butt to get access to. But it's a really nice paper. And I think they've done a great job bringing a lot of genomics techniques to bear on something where they've got a real biological question that they're trying to address. And they've used their genomics data to make a real advance in terms of how we think about all these E. coli subtypes. So I think that's a good place to start. Now, I'm by no means an expert, so there's probably a lot more to this than, and maybe Karen can comment, there's a lot more to this than we're getting from just the intro. But I think it's important to kind of give people some foundation or grounding on what are we talking about here with pathogenic E. coli. So it's Escherichia coli, which is an inhabitant of the human gut, but you can split it up into different types of pathogenic E. coli, and as I understand it, this is mainly done by presence or absence of some characteristic virulence determinants. So in this paper, they're primarily going to be talking about something called the locus of enterocyte effacement, which is a pathogenicity island. They abbreviate this as LEE. And Karen, it's said LEE, right? Or do people smash it together and say Lee?
Yeah, they call it Lee.
Lee? Oh, good.
Yeah.
Yay! That's going to make my life so much easier.
It's a Lee.
I was like, I really don't want to be saying LEE the entire time. So this Lee is a 35 kilobase pathogenicity island. It has 41 core genes that are organized into 5 operons. So we're looking at part of what people sometimes call the accessory genome of E. coli. So it's something that has to do with pathogenicity, something that has to do with virulence. And one of the things it encodes is a type 3 secretion system, which is the kind of little needle-like structure that bacterial cells can use to transfer effector proteins from the bacterial cell to the eukaryotic host cell. Some of those effector proteins are also in the Lee, as far as I know, but not all of them. So you do have some effector proteins that are encoded outside of this particular island.
The nomenclature makes it easy. So there's non-Lee effector proteins, which are NLEs, and then the other ones are typically associated with it.
Okay, so non-Lees and then Lees. it's like the first couple of paragraphs of this is just like alphabet soup. It's like, oh, okay, we've got EHEX and EPEX and TEX, and it's probably a good idea to kind of specify what are those that we're talking about. So what we've got here are atypical enteropathogenic E. coli. So they're going to call these AEPEC. And a regular EPEC, or a typical EPEC, is characterized by having this Li, so having this chromosomal pathogenicity island, and then also, as far as I understand it, having a something called the bundle-forming pilus operon. And this is a gene called BFP. And so your atypical enteropathogenic E. coli have the Li but don't have BFP.
Yeah. So the whole basis for this, this is all kind of from phenotypes. So EHEC is the diagnostic phenotype for a bunch of different infections. EPEC is a different type. The strains they're looking at are basically the leftovers that cause disease, but they're not quite sure how or why. Whereas the research before on EHEC, EPEC, that's where they come into like the bundle forming pilus and the Shiga toxin and stuff like that.
Yeah.
They're atypical because they're like,
yeah, and I think that's a good way to describe it, is to remind everybody that what we're talking about here is a combination of trying to link phenotype to genotype. And like you said, these were the ones, these atypical ones are the ones that they're saying, you know, they cause disease, but we're not entirely sure what is the genotype that makes it characteristic.
Yeah, they don't match the model of what we know right now. So
there you go. That's a good way to describe it. And I think that's one of, I don't want to jump ahead, but I think that's one of the big things that this paper and their approach comments on, which is if you treat these atypical enteropathogenic E. coli as one monolithic group, you're going to miss some of what's in their evolution that's leading to this particular phenotype. That's, I think, one of the biggest things that I pulled out of this paper, but we'll piece that together as we kind of go.
Can I ask a quick question?
Yes, please.
So if I took the only E. coli I ever work with are like DH5 alphas because I'm cloning some genes. If I put the LEE into the DH5 alphas, would it become a pathogenic, or is there more to the story than that?
No. So it would. There's more to the story. So there's a bunch of other things that change, plus the non-Li effectors, plus the Shiga toxin, which is a whole separate thing, or the BFPAs. It's a structure that enables pathogenicity, but it's not necessarily the sole determinant of the lifestyle, I would say.
Okay, and then if is the Shiga toxin, is that something that's secreted by the type 3 secretion system?
No, so that's typically, I want to say it's a type, I'm making this, it's either type 1 or type 2, but that's a complete guess.
Okay.
But yeah, no, it's a secreted toxin that disrupts a whole bunch of stuff. So.
Okay.
I'm not an E. coli expert, so this is my understanding.
No. I was just going to say, Karen, you were nodding a little bit. Do you have any further thoughts on that based on what your lab does? Yeah, unmute yourself first.
No, I was just agreeing with Dave, like with the fact that, you know, if you just take the Li and you stick it in the non-pathogenic coli, I don't think it will become pathogenic because of all the other things that are happening. But yeah, as for like whether or not the Shiga toxin and what it specifically does, I'm not sure as well. Yeah.
Okay. The reason I was asking is it's like the atypicals have the Li, and then the other two, the typical EPEX and then the EHEX, they have the Li plus an extra thing. And I was just wondering if the Li had to come first because I was thinking the Shiga toxin was maybe secreted through the type 3 secretion system, and so.
So through the wonders of Google, so the Shiga toxin is type 2, like a lot of the other like cholera toxin and stuff. There are effectors, so there are secreted effectors within the Li itself. I don't think any of those together are sufficient for pathogenicity, although they might be necessary for pathogenicity. And then I think the other point of this whole story is that this is the the cool part about this is that there's probably unknown other things in some of these atypical lines, because just because we know about the other extra allele stuff happening in the EHEX and the EPEX, there's probably something like that happening in the atypical lines, but we just don't know what to look for yet.
Yeah, that's one of the things I was going to say too. Is that I think exactly like you said. One of the big take-homes here is that we probably haven't characterized all the effector proteins that play a role in E. coli virulence, and that leaves us open for, you know, now that we can do whole genome sequencing, you can start to try to identify what might be some novel, common effector proteins that you're finding in these lineages.
Yeah, and this is so the challenge with that. So there are motifs that. With the type 3 secretion system, there's kind of this fuzzy motif in the N-terminus of the proteins that kind of tells the effectors to be secreted, but it's not great as a diagnostic of it. You can't just go through the genome and be completely sure of what's an effector protein just through sequence by itself. We're getting better, but we can only make pretty educated guesses, and then really the hard part is testing it in models to actually show translocation versus just secretion. And that's why, you know, we can, I mean, the other side of it too is that just because something looks like an effector doesn't mean it truly is in the genome either. So
yeah, that's true. Yeah. Okay. So
I had a quick question.
Yeah, please.
So are any of these, I'm not sure if the article really addressed it, but are any of these pathogenicity islands or virulence factors associated with antibiotic resistance outside of the plasmid, but I guess nuclear? Because different E. coli seem to be really selected. Like seems like a selectin pressure after antibiotics, or even some of the antimicrobials that I utilize, like berberine or oregano, things like that. So I kind of avoid using strong ones now. But in antibiotic studies, like Pat Schloss's study, there's always a bloom of E. coli after different kinds of broad spectrum antibiotics. So could the virulence be tied somehow tied to the antibiotic resistance?
So my understanding is that the lead does not itself encode any antibiotic resistance genes, although I could be wrong on that. Pulling back from what I did for my PhD work, one of the things that we were looking at was, in fact, we were looking at plasmids and how they moved around amongst E. coli and Salmonella, because those mobile genetic elements very commonly carry antibiotic resistance determinants. So I do think that there are some chromosomally incorporated antibiotic resistance genes, but you find a ton of them on the mobile genetic elements that are being carried on plasmids. And so
it's something we don't really understand quite well at any level now, but so I don't think there's any antibiotic resistance genes in these islands per se, but it's a type 3 secretion system that's going through the membrane, so that's going to feed back on all of the outer membrane responses and stress responses that way. So I could definitely see it being more tolerant to antibiotics, just because maybe efflux pumps or something get turned on more, just because it is kind of screwing with the membrane dynamics. But
well, that's true. Yeah. So there could be. It's not necessarily that there's evolutionary pressure related to them being encoded on the same element, but you might have some selective pressure for the actual mechanism or for the phenotype itself. That's true. Yeah.
And we're just terrible. We're terrible at understanding outer membrane efflux pump stuff. So.
Oh yeah, that's true.
I just wanted to add something really quick to what Laura was talking about the different plasmids. Like, particularly even just EPEC has multiple different plasmids, and there are virulence factors distributed on those different plasmids, and they are also very, very hard to purify because they're extremely large. So EPEC has a plasmid called the EAF plasmid, which contains some virulence factors, and my lab was looking at this, and at the beginning when I started in the lab, I actually had to try to purify the EAF plasmid, but because it was so big, I can't remember. E. coli was really incredibly big, and it was at such low copy that it was incredibly difficult for me to purify. Like, I would get multiple forms of the other smaller KB plasmids, like the 7 KB or the other ones, but the EAF plasmid was just, it wouldn't even enter the gel. It was so big. It was very difficult. So, yeah.
Are they enterotoxic toxins? These factors on the plasmid? Are they like not Shigella-like, but other kinds of enterotoxic toxins?
No, they weren't enterotoxic because we don't know if they were toxins per se, but we know that they are specific effectors. Like they do something to the host, and they change some of the cell biology within the host, and that is part of their virulence. But we were going to pursue it, and but that project didn't get funded, so we ended up dropping.
Oh, that's a compliment for that. So if anyone's watching this who makes funding decisions.
Thank you.
Okay. So does anybody else have any questions before we start to kind of launch into the data from the paper? I think these are all very good questions to get oriented about what we're talking about. Okay. All right. So what this paper is going to do is they've taken, they're going to try to look at evolutionary trajectories, population structure, talking about lineages and clones emerging. And they're doing this by taking multiple isolates, 196 isolates, from what sounds like it's an ongoing project where the sampling sites appear to be primarily in Africa and Asia. And they're going to combine those. They do whole genome sequencing on those. And they're going to combine those with 171 publicly available E. coli genomes. So this gives them a pretty big set of genomes to work with, which is great because they've got a lot of information to try to look at genetic variation and to try to kind of tease out how they're all related to each other. So they got 359 E. coli genomes in their data set. And I'm going to see, let's see, this is the first time I've ever tried to do real people screen sharing rather than just holding up the paper. So we're going to see if this works. So do you guys see that? Does that look all right? Okay.
That's a figure.
Cool. So you're like, it's a figure. So this is their first figure where they're looking at the, they've got a phylogeny which is based on core genes. So right now they're not looking at the accessory genes. They're just going to look at core genes that are found in all of the genomes, and they're going to make this phylogeny on the basis of the SNPs. So that's how they've kind of clustered all of this stuff. And they've got them marked by their type in terms of whether or not they're EHEC, EPEC, atypical, EPEC, pick your alphabet soup that you want.
Just to chime in as a random aside to this, if you guys ever, like, Kat's webpage is awesome about the pipelines and just, like, computational genomics for beginner stuff and a nice following directions of things. So if you ever stumble into Kathryn Holt's website, it's kind of really good for genomic stuff from kind of beginners to advanced too. So it's just a
Does
good
it kind of walk through like working with data like this? Is that part of what the?
there's parts of it, yeah. There's an awesome document about. I'll find it and then I'll post it back up here.
Okay.
You continue.
Okay. Yeah. So I think that's great because I think this is the kind of data. I mean, so many of these biological questions, you can do a lot if you've got this level of data, as long as you're asking the right questions and you have the skill set and the knowledge to address them. So that's certainly a helpful resource. Okay. So what are we looking at? What does all of this mean? Basically, what this told them was that they identified out of this 10, what they're going to call clusters or clonal groups of the atypical enteropathogenic E. coli. And they've got them labeled with the CG here. And so they're showing you kind of in these different colors which clades on this particular phylogeny they've pulled out as being separate phylogenetically distinct clusters. So this kind of becomes important because they're going to be talking about how these clusters vary, and they're going to be talking a little bit about what might be different or similar amongst those clusters. So that's that first figure. I don't know if anybody has any thoughts or questions about that first figure.
I was, like, I think I was confused in terms of what are the colors in the middle versus what are the colors on the outside of the. I know the colors on the outside of the ring are the different types, the pathotypes that those would belong to, like in colors, but I wasn't quite sure what the inner color represents.
So the outer color, you're right. The outer color is the what they're calling the pathotype. So is it EHEC, EPEC, atypical, EPEC? and then they've separated them between the isolates that they sequenced from their own sampling sites versus the ones that were publicly available. But then the inner color, I think, like for instance, this dark purple, I don't know if you guys can see my arrow, but the dark purple cluster here, I think that's just a color that they picked to distinguish this clonal group 517 from, for instance, this clonal group 40. So they're highlighting exactly which of these endpoints, which of these genome sequences they're including in each of those clusters, and they're trying to differentiate them with these different highlighted colors.
Okay, so the inner circle and the outer circle aren't necessarily related in terms of something that they're trying to tell us. It's just they're trying to differentiate between the different, did you call them clades?
Yeah, I mean, in the phylogeny, you'd call these clades. They gave them a kind of a term that they're going to call them either clusters or clonal groups.
Okay.
And you know, so this section that we're looking at here, that's kind of the light green. There's a whole group here that's a lot of atypical EPEC from their sampling sites, but for whatever reason, it looks like they did not choose to include all of those in this particular clonal group that they're looking at. So that's probably done on the basis of the SNP analysis. They're going to pull out for a certain threshold of similarity and group things together in a clonal group.
So do I have it right that the clonal groups are all similar in the fact that the Li is inserted into a specific site in the genome, and so like within a certain...
Not yet. So
I'm jumping the gun.
Yeah, you jumped the gun a little bit. So this figure is looking at core genes only, and looking at single nucleotide polymorphisms within those core genes. So the way that they have identified the clonal groups at this stage does not incorporate any information about virulence determinants or really anything from the accessory genome.
I mean, just anytime you call something a clade or a clonal group, it's still kind of subjective.
Mm-hmm.
You have to make some reference to where you're going to actually, in the tree, call it a clade or a clonal group. So, you know, based on sampling and whatnot too. So it's, yeah, there's some, as they go into it later, there's some diversity, at least in the insertion points for the secretion system, even within the clonal groups that they call them.
Yep, absolutely. Absolutely. Yeah, so, and I mean, one of the things, I might stop screen, I really don't mind actually not being on the screen and having this up instead, but I'm like, hi, I'm back. One of the things I noticed with this that I had put a question about was that they said 4 of the atypical EPEC clonal groups contained isolates with additional virulence factors BFP or STX. So if you remember, the STX is the Shiga toxin, and the BFP is that bundle-forming pilus gene. Both of those are virulence determinants that are typically characteristic of a different pathotype. So when they put that sentence in there, I'm guessing what that means is, you know, they're gonna talk about this later, but that you're seeing atypical EPEC with its characteristic genotype grouped in with EPEC that maybe appears to have acquired another virulence determinant that now shifts it into a different category. So everything seems very fluid at this point. You know, it's one of those things where it's like we're trying to put things in boxes, and we just like to have lots of order about how things work, but evolution doesn't always cooperate with that.
I'm not completely sure where your sentence came from, but what I think I understood was that these were all categorized as atypical because they did like a PCR screen, and then when they were negative for the STX or the BFP, and then when they did the whole genome sequencing, they did find those genes there, so they just had to recategorize them later, drop them out, and call them the EHEC or the typical APEC or EPEC.
Yeah, that's certainly true that they've got, kind of prior to the place that I was talking about, they talk about how having by the advantage of having whole genome sequences that you can get more precision about whether or not you can detect a variant of a gene like BFP. I think even with that though, it's a little bit later. It's kind of in the second column of page 3, a couple sentences down. It says for the AEPEC clonal groups also contained isolates with additional virulence factors, BFP or STX. And so I think those are going to turn out to be legitimately a different pathotype, perhaps, that that end up clustered in that clonal group. So that tells you something about the evolutionary trajectory of that particular lineage. Okay.
But I think in page 3, the left column 2, so one of the BFPs is there's a divergent allele of it, which is why it might not have shown up in the PCR screen, right? So.
Yep.
Genome analysis revealed the presence of a BFP operon with a divergent form. So, I mean, whole genomes versus just kind of targeted screening.
Yep, definitely. Okay. All right, so they've got some sense of, I mean, that's kind of looking at vertical evolution, as they call it. So you're not really looking at horizontal transfer at this point. And then what they're going to do is they're going to talk a little bit about how they find these clonal groups in these atypical enteropathogenic E. coli. And so that leads us to their kind of interesting representation of this. It's like their 3D stacked bar chart on top of a map, which I thought was kind of cool. So I'll see if I can pop that up there. So there's that. They have a rarefaction curve, which they say suggests that they haven't actually kind of reached saturation with finding different clonal groups. And then they're just looking at, Karen, go ahead, yeah.
Oh, I was just going to ask a silly question. What is a rarefaction curve?
Ooh, Grace, what's a rarefaction curve? We can answer this now because we went to the mother workshop. Go, unmute yourself. You have to unmute yourself first. We can't hear you. That's okay.
Oh, yeah, sorry. No, I can't do justice to what I learned, so I think you should go. But you take out data. So you can compare a sample of 10 with a sample of 300, and they can look even. That's all I know.
Yeah. So what the rarefaction curve can do is what it's supposed to kind of model is, so they've got here the number of the isolates that were sequenced, and then the number of the lineages discovered. And so what you should see if you have, basically what it's telling you is, if you continued sequencing isolates, would you continue to discover new lineages? And if that were not true, you would kind of reach a plateau in your curve where no matter how many sequences you added, that curve would not continue to go up on the y-axis. But what they're showing here is it looks like you would still, like if you extended this curve from South Asia, it looks like you would still continue to see an increase in the number of lineages discovered if you kept sequencing isolates. So the rarefaction curve is kind of telling them, if we sequenced more, would the y-axis data point continue to rise versus would it just plateau and you'd get to a point where it didn't matter if you sequenced 1,000 things, you still wouldn't find any new lineages.
So that would be part of why they were talking about with the West Africa samples, that the fact that they weren't able to categorize it as well as the other ones is because they didn't have as many isolates.
Yeah, absolutely. Yeah, you're right about that. And you could kind of see that reflected down here with the timeline that they did, where they kind of mapped some of the 10 clonal groups and looked at kind of temporal stability. Do they continue to detect that clonal group over time? And some of their data points for West Africa are pretty short. And as Karen said, they commented on, they don't really know if that means that it's very transient or they just had a small sample size for West Africa. You could kind of see when they're looking at the map, they've got an N of 13 for the Gambia, they've got an N of 33 for Mali. So in terms of looking at West Africa, that might not be enough to work with in order to say anything about that.
I have a geography question. Is it really called the Gambia, or is it? How come that has a modifier on it?
I think it is called the Gambia. I don't want to say for sure, but I think it's true. That's a good Wikipedia question, I think.
The Gambia is a country in West Africa, mostly surrounded by Senegal, with a short strip of its coastline bordered with the Atlantic Ocean at its western end. So yeah, the country, it says Gambia, but they do call it the Gambia in Wikipedia.
I think that's excellent. I think everybody should get to put a modifier in front of their country.
One of the Dakotas needs to declare themselves.
Dakota.
Yeah, the Dakota.
Oh, that would be a great move.
Yeah.
Get on that, Dakota.
a lot of universities that are University of Montana or University of Ohio, they say the University of Montana, and they actually make it part of their school name. So
I'm familiar with this from the Ohio State University. They're very serious about that over there. All right, so we've got here's our sampling sites. We've got samples from Africa, samples from Asia. And they're kind of just looking at the different clonal groups that they found and the distribution of them across these sampling sites. They also look a little bit, as we said, at temporal stability. So in South Asia, it looks like, you know, quite a few of these seem pretty stable over time. They're detecting them across 3 years, which I think is another really interesting aspect of this, is looking at how these things across a time series, if you continue to detect these clonal groups. Okay. So that's figure 2, and then what they're going to do is they're going to start delving into looking at the kind of the evolution and the population structure of the actual Li. So up to now, we've been kind of looking at the core genes as a way of getting oriented with how these lineages and isolates might be related to each other. And now they're going to start looking at the actual locus of enterocyte effacement, and they're going to look at nucleotide variation. And I'm glad Dave is here, because I have a question for Dave, which is this an appropriate context for using dn over ds? Or are we worried because this is kind of within a population? Does dn over ds actually mean something here?
I think they're using it within the clades, though.
Okay.
So it's kind of they're looking at it from the vertical inheritance.
Okay.
I think I read it quick, so.
Okay, so they're looking at, and I'll pop this up, so they're looking at 2 different things here in this figure. And their goal here is to look at, as they say in the paper, genetic variation across the full complement of 41 Li genes. So what they're going to say is 2 different measures. Over here on the left, they've got nucleotide similarity that goes from 0 to 100%, and that's the box plots. Over here on the right, they're looking at something called dn over ds. So what this is intended to be a measure of selective pressure. It's the number of non-synonymous changes per non-synonymous site over the number of synonymous changes per synonymous site. So if you have more non-synonymous changes than synonymous changes, then that suggests that your sequence is under positive selection. So if dn over ds is greater than 1, then that suggests strong positive selection. If dn over ds is less than 1, that suggests purifying selection. And if it's around 1, usually people take that to mean neutral.
That's all completely arbitrary.
Yeah, I know. I was just going to say there's a lot of theory in molecular evolution that talks about the values of dn over ds, when you can apply it, what it means. So take that with an enormous chunk of salt. But
just to clarify something you said there. So the dn/ds is the counts. The ka/ks is taking into account the number of sites possible.
Okay.
So you'll see those 2 ratios, and they just basically, it's dn/ds is a cruder measurement than ka/ks, just because it's not necessarily taking into account all the maybe skews in the amount of sites that could change. But
okay, so that one's not. So dn/ds is the one that's not corrected for sites. That's just the
yeah, I think that's the one that's.
Okay, so that gives you a measure of selective pressure. And that's the dots here. So we've got each gene of the I don't know if this is every gene or maybe a selection of the Li on there, and then they've got nucleotide similarity up here, and they've got the measure of selection down here. You know, I'll be perfectly honest with you. I looked at this and I was like, I don't really know what I'm supposed to get out of this. It's neat data, and I understand why they did it, but just looking at the figure, I didn't come away with a really strong take-home message from the figure itself. Did anybody else have any thoughts about this particular figure?
I just look at it in terms of variability, so you can see where the more variable portions of each are versus the more conserved ones. But to read any more into it specifically is kind of stretching things, I would say.
Okay. Okay. So I think this is where they start to build an argument that the type 3 secretion system genes, which are shown here in their formation as a kind of protein structure, are more conserved than the effectors. And I can kind of see where they're getting that from. So those genes are in purple, and in terms of nucleotide similarity across these genomes, they look pretty conserved, while you've got the effectors that seem to be a little less conserved in terms of sequence.
Yeah, I mean, and that's kind of what you see across a lot of these secretion systems, because you don't expect the structural genes to change too much because they have one function within the bacterial cell. Parts of them might be recognized by as a PAMPs or something as by the innate immune system, but the effectors are the proteins that are actually getting translocated into the eukaryotic cells, and so that's probably where more of the kind of arms race interactions are going on there. So, I mean, it's kind of what you see across a lot of these systems.
And also on their picture of the structure of the type 3 secretion system, the part that contacts the host's cell membrane, those are ESP B and D, and so those are ones that had more changes or less conserved. So there's some positive selection maybe.
Oh yeah, I see what you mean. So you're talking about these here, and then we've got A, and then A's right there, and then over in our graph we've got here's A, here's D, and here's B. And those are some of our, as you said, Joel, those are some of our less conserved in terms of nucleotide similarity. And it does look like their DNDS values are on the highish side for these type 3 secretion system proteins. All of the DNDS is less than 1 though, so I don't know. I don't know what you can really say.
It's hard because you only need one site to be selected, and there might be a ton of other non-select. Selection just means that at least one site is positively selected for, right? So there can be a lot of other synonymous or non-synonymous changes there.
Yeah.
That obscures one site or more, but it's route 1 is just kind of a crude measurement of it all.
Yeah, and I don't want to get too into the weeds on this, but I'd read a paper, I think it's from like 2008 now, by Joshua Plotkin and the first author. I'm not even going to attempt to pronounce his name, so sorry. But they wrote a paper about kind of the population genetics of DNDS, and they kind of made an argument that it sometimes gets used inappropriately if the evolutionary distance is not large enough, so that like within a population, they kind of showed that DN over DS doesn't really follow the assumptions that you would expect, like between species, for instance.
Yeah, plus Josh has another paper showing that synonymous changes matter too. So
yeah,
I
mean, everybody uses it because it's a good measure that people who do this kind of molecular evolution recognize. But after I read that paper, I've always wondered, I wonder how much DNDS over DS is getting abused.
So I always look at it in a relative way. So we've got 40 genes here where we can kind of compare values, and we can see these genes are more diverse and they have different DNDS values than the ones at the baseline. And that's kind of how, you can't necessarily show, this doesn't demonstrate selection on them, but it trends in the right direction because you kind of have the control there for the ones that aren't changing as much as the ones that are changing a lot. So.
That's true. That's true.
Sorry.
Okay, I was just going to say, if we ignore the DNDS and just look at nucleotide similarity, it's more clear there.
Yeah, that's true. Karen, what were you going to say?
Oh, I was just going to add something unique in terms of from the host perspective, what's special about EPEC and EHEC, or specifically enteropathogenic E. coli, which is that the tier and the intamen that you're looking at on the right side with the actin arrangement. So what happens is the E. coli eject their own host receptor into the cell, and that's tier. So through the type 3 secretion, tier is ejected into the host cell. it's displayed on the surface as a membrane protein, and then intimin, which is the ligand, binds to TEER, and then it initiates intimate attachment, which is why it's called intimin. And so then from there, the type 3 secretion system starts ejecting these effectors that create this actin arrangement, and it raises the top of the cell where the bacteria is on top, and this actin arrangement is called a pedestal. And so the bacteria kind of sit on top of this little pedestal, actin pedestal, and they don't really know what the pedestal is for. Like, I've asked multiple times, what is the pedestal for? But there is a theory where bacteria kind of surf on top of the pedestals, and it helps them be transferred from cell to cell. But what I found was unique about the figure is that TEER, which is, as we were mentioning on the host side, is incredibly, in terms of the nucleotide similarity, is incredibly variable. But intimin on the bacteria side is incredibly stable. So, yeah, I mean, like, I definitely got that there's more variability on the host side, but I just found that it was strange that if TEER is variable, then why, I guess it would be interesting to see where that nucleotide variability is. Like, it couldn't be in the place where it binds to intimin, because then intimin would also be variable. So then that nucleotide dissimilarity must be on the host side.
Oh yeah, that's interesting. So here's what you're saying with the part of the figure that shows here's TEER and here's intimin, and I see exactly what you mean here. So here's TEER in terms of nucleotide similarity, and as you said, Karen, this is one of the ones that looks like it has quite a bit of variability at the sequence level, and there is intimin, which looks a little more conserved. So yeah, you're right. I think it would be really interesting if you were to look at that sequence and start to kind of break it into regions, whether or not the regions of the sequence that lead to parts of the protein that interact with intimin are more conserved. Mm-hmm. That would be really interesting. Cool. Okay. Yeah, thanks, Karen. That was I don't know anything about that, so that's a good perspective on that.
I mean, anything called like enterocyte effacement, it just sounds so toxic and lethal.
Yeah.
And I think understanding, this pathophysiology part helps me too.
Yeah, and that's what they're saying down here is that's I guess based on what you were saying, that's when you get a lesion. Is that part of what the pedestal you were mentioning? Does that contribute to getting lesions?
So that it attaches and effaces. I think. Don't quote me on this. If I remember, based on pili, like it attaches and then somehow it actually effaces the microvilli of the intestine. So that, like the actual TEER intimin part, aren't the ones that are responsible for the actual effacement of the microvilli, but that happens before the intimin attachment occurs.
It's more related to what's the effectors, right? The chemicals being secreted?
Well, the type 3 secretion ones that are being displayed here aren't secreted until after the attachment occurs. So first you have, if I remember, there's this great video on YouTube where they've done this HHMI video, Howard Hughes video, where there's pili that kind of anchor the bacteria into the microvilli, and then as the bacteria is hanging out there, the area where it's about to attach onto get effaced. So the microvilli are effaced. As to how that actually happens, I'm not particularly sure, but that event happens before the intimate attachment or even the type 3 secretion effectors that we're seeing here actually get secreted.
Okay. I see. Did somebody Dave, did you just find that and pop that up? Somebody did.
I think so.
Okay. Cool. So if you go into the chat, you should be able to see the link for that. I think Dave might have found that video Karen was describing.
I mean, in a way, this sounds kind of like a zit, but on the microvilli. And it's kind of funny because when I have clients, like if they complain of acne at some point or skin cysts or something, it's funny because their gut will have like much higher levels of staph or strep in the gut versus normal controls. Or if you look at 16S gut microbiota studies for skin and comparing with gut microbiota, and I always want, and there's always E. coli present as well, higher than normal. And I wonder if all of these share some kind of similar pathophys in kind of melting or emulsifying the microvilli and gaining entry or somehow causing the disturbance and leading to dysbiosis.
Oh, yeah, maybe so. Yeah, there's a lot of connections that I think having more of this data will help us start to uncover.
Yeah. We'll have to radio label some of this and see where it goes.
Yeah, there you go. Okay, so so the other thing they wanted to do, so they've got this data on variation across the Li. The other thing they wanted to do with this data, I thought was actually a really neat idea of trying to think about how these genes might be co-evolving together, or if they're co-evolving together. And so what they did was they made individual gene trees. So imagine for each of these gene sequences that are shown down here, they took, you know, all of the gene sequences for intamin, for instance, and they made a phylogeny of those gene sequences, and then they made another phylogeny of the tier genes, and then they made another phylogeny of, you know, ad nauseum. They did these phylogenies. Some poor grad student or postdoc sat down and did all of this. And then what they did was they looked at network analysis so that they could try to figure out, looking at those phylogenies, which of these genes might be co-evolving together. And so they came up with 4 subnetworks, 2 of which actually have quite a few genes, 2 of which don't have that many. And then they've got 9 singletons. So the main thing I got out of this was they're looking at these 2 subnetworks, subnetwork 1, subnetwork 2. They have quite a few of the type 3 secretion system genes in them. So this subnetwork 1, it looks like there's type 3 secretion system genes co-evolving together, a few regulators and a few chaperones that are co-evolving together. And then you've got intamin and tier kind of off on their own. And then you've got your effectors, with the exception of this one that's in this network. Most of your effectors are, you know, singletons, and you've got one that's in a little network with another gene. So this also, I think, is contributing to what they're saying about that the type 3 secretion system is more conserved, the effectors are evolving a little bit at a higher rate. Okay, so then they're getting into looking at kind of trying to figure out the different lineages. And this is where they start to identify that within atypical enteropathogenic E. coli, they've got a couple of different lineages that have followed different evolutionary paths to get to this point. And I think this is probably one of the big strengths of having all of this whole genome data, is you can really start to piece this together. So what's going on here? Okay, well, they've got lineages here. So this is based on a phylogeny of the Li. So this is a phylogeny of the locus of enterocyte effacement. They've made this phylogeny, and they're showing 3 different lineages, one of which contains quite a few of the subtypes, and then 2 that have fewer subtypes. They also start talking about the insertion site, which I think a couple people have mentioned at this point. So there are 3 places in the E. coli chromosome that they see this Li inserted. All 3 of them, I think, are tRNAs. And what they're showing here is proportionally where was the insertion site for the subtypes that are shown here. So for the 9 isolates that are within Li subtype 1 in lineage 1, how many of them were inserted at which location? In this case, it looks like all of them were inserted at the tRNA for phenylalanine. Then what they've got over here is they've started to look at the effector genes that are, is the N, I can't remember why it's NLE. Does anybody remember why that's the notation they've used there?
Non-Li effector.
Non-Li, thank you, yes. So what they're showing here are effector genes that are not present in the Li, and they've got a heat map that basically says are they present or not present in terms of the number of isolates. So I think if it's present in all of the isolates, it's going to be really dark red. If it's not present in the isolates, it starts to kind of fade to white, depending on how many of the isolates actually have that particular gene. So that's a lot to pack into one figure, but I think it's an interesting way to start to look at rather than thinking of atypical enteropathogenic E. coli as a big monolith, they're starting to kind of split it out into these different evolutionary trajectories and starting to kind of try to get a way of thinking about them in terms of these different types. So they're going from the big scale, which is the lineages, down to the subtypes. Yeah, I mean, what did everybody else think of this figure? Silence.
I found it. Sorry, go ahead.
I was
Joel.
just going to say, this is one where color would have helped me out.
Yeah, probably so. That's true.
I was just thinking. I was just going to mention that where we see that EPAC, where it says the plus, where it says contains EPAC or EHAC, those ones seem to have more of the broad range of the non-NLEs compared to the other atypical strains.
Oh, that's true. I see what you mean. So these notations, well, that's got our outgroup in it, but not that one. But the stars and the pluses are subtypes that appear to include typical EPEC and also EHEC if there's for the asterisks, or pluses typical EPEC. Yeah, you're right. That's true. It really does look like those might have a larger number of these non-Li effector proteins. That's interesting.
But again, that might be a sampling thing where we know that they're non-Li effectors because they were found in those kinds of things.
That's true, and that is a point that they make in the. I think they make that point in the text where they basically say, you know, all this is, it's like microarrays, right? Like if you know about it, you can put it on a microarray. If you don't know about it, you're never going to find it with a microarray because it's not on there on a probe. So the same thing holds here. We know about these because they've been described as effector proteins, but that doesn't mean there isn't a whole panel of effector proteins that have yet to be discovered that's missing from this particular figure. That's a really good point.
So in terms of evolutionary origins, like we wouldn't be able to determine that simply just from the presence of the different types of NLEs, because the NLEs might be in such a degraded form in the atypical or not recognizable or won't bind to the probe.
I think their argument is that there may be non-Li effector proteins that just aren't in this list. That these are the ones that have been recognized and described, and it should continue on out this way with entirely new open reading frames that have not been characterized as effector proteins yet. That was what I took from the text, which, you know, I don't think is all that surprising. I'm sure there are effector proteins that have yet to be described.
And to that point, though, I think the most, just kind of glancing quickly, the most interesting comparison to me is 9 versus 10 in lineage 3, because you have, seems like a complete switch.
Oh, yeah, that's true. There's 9.
That one, it kind of just, they're inverted for some reason, but...
Yeah, so there's 9, there's 10. So you can see 10 has quite a few of these described effector proteins that are not in the Li, and then 9 just doesn't seem to have many at all. Huh. Yeah, I mean, I don't, yeah, it's interesting how all these got acquired and what steps those took. You know, that's hard to reconstruct, I think.
But it looks like it's almost, I mean, just parsimoniously looking at this, if you look at 10, 11, 12, they're kind of the outgroups for lineage 3, and so maybe that's the ancestral state. 9 kind of lost them, and then 12 through 30 kind of lost them independently.
Yeah, that's true. So you could track that back to if you're looking about losses, comparing this clade. You've got a clade here, you've got a clade here, and then all of this is a clade, which if it experienced in the common ancestor some particular evolutionary events like gene losses, you'd see that reflected in all the descendants. Yeah. So some of these conclusions are things where I think that the concept phylogenetic independence kind of comes into it. So do you have lineages that you can explain these changes because of actual evolutionary events that happen to each lineage, or is it that you've got a sampling of things where what you're tracking is basically something that happened to the common ancestor way back when, and you're just seeing it reflected in all the isolates you got?
And this is where it's nice, where there's multiple isolates within each within the tails of the phylogeny here too. So it's not just like typically you'll see these and it's like one strain here and one strain here, but here's with the heat map, it kind of represents the variation within that those specific nodes and clades on the tree, which is
that's true. Yeah, that's true. This is a really good data set for all of this. I mean, and that's what makes the insertion site data really interesting too, because what it's telling you is that, for subtype 1, 100% of these had the same insertion site. But you can also see that there are some which 8 over here has quite a few isolates in it, has 71, and you start to see the effect of, I think, maybe greater sampling, because you're seeing that, I think this might be unknown, but you're seeing 2 other insertion sites kind of represented proportionally.
It makes me wonder how they're actually moving around, though, because that seems like I don't think there's a clear explanation for, we know why things get inserted in certain places, but to actually figure out the intermediate steps is really hard, and it's hard to think about. But.
Yeah. I mean, when I was doing work with plasmids and transposons, it was like, that was one of the things I was frustrated by, is that I didn't have a good sense of the mechanism of how things were moving around. You just saw it after the fact. You just got this snapshot of evidence of stuff's been moving, and it was really hard to reconstruct exactly how that might have happened. Yeah. So then what they're doing is they're going to take this information and they're going to actually look at the distribution of the subtypes and the insertion sites across the 252 E. coli isolates. So we're back here to thinking about the core chromosomal phylogeny again. So this phylogeny, in contrast to the one that they're showing in the inset, that's the phylogeny we were looking at just previously. That's based on the Li. This phylogeny we're looking at now is actually based on the chromosome, on the core genes. So they're looking at, there's Escherichia albertii as our outgroup, so all of these other ones are E. coli. And then they've got mapped on there what the Li subtype is in the number, and then the color of that block tells you which of the major lineages it belongs to. And then they're doing the same thing where they're showing the insertion site, and so I think they're just trying to get a sense of looking at how the core chromosome has evolved and trying to map what they know about this accessory genome component onto that now. Yeah, and I mean, this figure just contains a tremendous amount of genomics data, so I think it's a reasonable way to try to present all of that, because that's just a lot to get into the figure. But it's interesting that we've got, one of the things I thought about this was that it was interesting that down here in this part of the tree, we start to kind of get some clonal groups that have like a mixture of the major lineages. So that's what it looked like to me, which I thought was a little odd. So I think they're just trying to get a handle on reconstructing the chromosomal evolutionary trajectory and the accessory gene evolutionary trajectory, which aren't always the same thing. So.
It almost looks like what you were mentioning before, Laura, that the sampling creates more variation, especially with those, with the ones that you're looking at down here, because that N is representative of the amount of isolates they have, right?
Yeah, that's my understanding. Yeah, you're right. It does look like, so these ones that I was pointing out are ones where they've got quite a few isolates. Which is not necessarily the case for some of the other ones, which they've only got one isolate for those. So I'm sure they're working hard to get more samples, because that'll help them clarify this a little bit more. So. Okay. So, I mean, overall, I think they got a lot done with all this data, and I think one of the things that they're showing with this, it gets back to the original point we were talking about at the intro, which was basically, you really can't lump all of these atypical enteropathogenic E. coli into one big group and hope to try to tease out, what makes them atypical. What they're finding here with having all of access to the genome data is that, each of these seems to be following a particular evolutionary trajectory. You can split the Li into 3 lineages, you can split it into 30 some odd subtypes, and then you can split the chromosomes into 10 clonal groups. So trying to map all of that variation together, just the big take home for me at least, was that you're going to, answering why these are virulent and what determinants make them virulent is going to be specific to each of these kind of groups. And trying to tease it out that way, which I think is a good way to use this data. What did you guys think of now that we've worked our way through all the chromosomal phylogenies and Li phylogenies? What did you guys think of the overall their take-home message?
Well, I guess my take-home is that at least they now have a roadmap how to start characterizing these, and so you were just kind of saying that, but trying to figure out how they're pathogenic, you can now subdivide into different groups and start to understand a little bit better. So, that's my understanding. This is probably going to be a paper that's cited over and over as these different groups get kind of characterized and figure out what made them pathogenic to begin with. I was going to say one other thing. David had put some links over in the side, and I think Karen maybe wrote the
little write-up,
the blog post or whatever. Yeah, and it had a link to this Kathryn Holt's website, and the link that David put up is a beginner's guide to beginner's guide to comparative bacterial genome analyses. And I had kind of taken a look at this before, and they have a more, as a supplemental data, they have a more step-by-step description of how to do all this, and it's a fairly long PDF. I had tried to get that before, and like a month ago the link didn't work, but it seems to be working now. So I do think that when I saw it, I was like, oh no, it's that thing that the supplemental data's link is dead, but it seems to be working now, so it might be worth taking a look at.
Yeah, it sounds like there's a lot of, that this lab has produced a lot of helpful resources, not only the data sets, but also some guides about how to kind of work your way through data like this.
And she's really responsive on Twitter, too, if you have any questions. Like, she answers questions all the time, so.
A good resource.
Yeah. I will say the one thing I want to end on is the figures 6, at least. They're working all with pathogenic isolates, and so these are things that cause disease that came into clinics at some point, right? So that doesn't necessarily mean that there's not non-pathogens that aren't sampled, that aren't in that tree. That could help to kind of elucidate things too.
Yeah, that's a really good point. So, especially when you're talking about the core chromosomal phylogeny, it would be interesting to put in some isolates that are not pathogenic to kind of see where they fit into the scheme of that.
Yeah, I mean, they might not exist, but they might exist in this context. I don't know.
Yeah. I mean, I really liked this paper. I thought this was. It's really nice to see. As somebody who's kind of struggled through various genomics projects, it's really nice to see somebody use a genomics data set to really say something significant about the biology of their study organism. Which is, what everybody's trying to do, so I think everybody's headed that way. But they really took a big data set and broke it down into some very logical components about trying to get at this question of what's going on with the evolution of these atypical pathogenic E. coli. So I thought this was great, and I think whoever did their data visualizations probably worked really hard on that, and you could see it reflected in their figures. I think those are well-chosen figures for the type of data they're trying to present.
And somebody at some point pointed out the Gambia, not just Gambia. So paper development at some point. That happened at some point. I'm guaranteed.
And we too have learned today that it actually is the Gambia. So we leave this educated on more than one front. Okay, well, does anybody have any final thoughts about the paper? I know we're getting up onto 4:15 here, which is going a little long, so I don't want to keep people too much longer.
That was awesome.
Yeah, thanks, and thanks to Grace. I think this was your first time joining us, so thanks for
Thanks for having me.
Hopefully,
I learned a lot.
And so I think we've already picked our next paper, which is going to be on bacterial endosymbionts, which I'm super excited about because that's what I did my postdoc on. So,
oh, yay.
In terms of talking about trying to say something sensible about genomics, that's what I spent a lot of my time doing in my postdoc. So to varying levels of success, I think. But we'll get that posted soon, and so that'll be next month. So we'll be back in March, and hopefully everybody can join us then. But thanks everybody for being here today, and have a good week.
Great, thank you.
Bye.